Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullah. Our third lecture for the immunology is going to be about antigen structure, processing, and presentation. So the objectives for this lecture is going to be define some of the terms that we're going to come across during this course, such as the antigens, epitopes, types and sources of the antigens. Also, we should be familiar with the antigen processing and presentation. Also, the role of the major histocompatibility complex in the antigen presentation, and discussion of the role of the antigen presentation in the generation of immune response. So definition: antigen is any substance that causes immune system to prompt the generation of antibody. Antigens can be proteins, can be polysaccharides, conjugates of lipids with proteins, lipoproteins, or they can be glycolipids as well. So the antigen can be a foreign substance from the environment, such as chemicals, bacteria, viruses, or even pollens. Another terminology we're going to come across is an immunogen. Immunogen is a stimulus that produces a humoral or cell-mediated immune response. When we talk about an antigen, it causes immune system to prompt the generation of antibodies. So it's going to activate the humoral immune response. An immunogen is a stimulus that produces either a humoral or cellular immune response. Another important terminology is the hapten or the haptens, which are low molecular weight substance. They are low molecular weight substance. By themselves, they are not immunogenic, so they cannot elicit an immune response by themselves, but if they were coupled with a larger carrier molecules such as albumin or globulins, then they become immunogenic. And to get a clear picture of that, so this is these pink triangles are the haptens, and here we have the albumin or the globulin carrier. So the hapten by itself does not produce antibodies. They are non-immunogenic by themselves, so there is no response from the immune system towards them, so there is no formation of antibodies. The carrier itself is immunogenic, and it is going to produce antibodies against the carrier itself. Once conjugated the hapten with the carrier, then this is going to lead to production of antibodies against the hapten and against the carrier. But mostly antibody production is going to be towards the hapten itself. So hapten alone, not immunogenic. The protein carrier is immunogenic. Once conjugated, it's going to produce antibodies against both the hapten and the carrier. But mostly, it's going to be concentrated, and most of the antibodies are going to be targeting the hapten and not the carrier itself. An antigen may also be formed within the body, as with bacterial toxin or tissue cells. So once the bacteria is introduced into the body, it's going to produce toxins. These toxins might serve as antigens. When we talk about the Clostridium tetani, Clostridium botulinum, those are toxins which are produced by the bacteria, and they serve as antigens as well. Or the tissue cells, such as the cancerous cells. Properties that make molecule more effective against antigens. So for a certain molecule, in order to be highly antigenic or highly or produce an immune response against, it needs to be foreign. So self-antigens are recognized by the immune system and they are not reacted upon. If the antigen is foreign, then it's going to be recognized, and it's the immune system is going to respond to this foreign antigen and produce immune reaction. Stable molecule that assume and maintain a definitive shape. So if they have a definitive shape, عندهم شكل معين مش متغير، كل ما كان الشكل ثابت بيكون more immunogenic أو more immunogenic. Larger molecules with molecular mass between 5,000 and 100,000 Daltons are more effective antigens than smaller molecules. Molecules that are structurally complex with distinctive shape and novel subunit combination. If the molecule is complex with distinctive shape, عنده شكل معين وعنده subunit combinations, each of these subunits might serve as an antigen. Route of administration. Usually molecules administered parenterally through the IV route are more immunogenic than molecules which are administered orally. So these are the 5 properties which make any molecule a more effective antigen. Antigenic determinants or epitopes. The body recognizes antigen by the 3-dimensional shape or region called antigenic determinant or epitope, sites on or with antigen with which antibodies react. So what is an epitope? Let's look at this molecule. This molecule is considered an antigen. This part of the molecule is considered an epitope. So it's the region of the molecule that interacts with the antigen binding region of the antibody, which is called paratope. So this is epitope, and this region is called the paratope. This molecule might have more than one epitope. So this area serves as first epitope, this area serves as second epitope, this area with smaller protrusions on the back also might serve as a third epitope. So a single antigen might have multiple epitopes. 2 types of antigenic determinants. We have conformational determinant and we have sequential or linear determinant. And we need to look at this picture in order to understand the difference between the linear epitope and the conformational epitope. So in the linear epitope, they are mainly recognized by T cells, but some also may be recognized by B cells. It's mainly recognized by T cells, but in the linear epitope, the antigenic region is the antigenic region in the linear epitope comes in a linear sequence or following each other, as we can see here. So this is the epitope. If the molecule or the antigen is denaturated, we can see denaturation of the antigen in the linear sequence. is not going to affect the antigenicity or immunogenicity of the sequence because it is in a single region. While when we talk about conformational determinants, then we're talking about amino acid residues that are not in a sequence but become spatially juxtaposed in the folded protein. They are recognized by B cells or antibodies. So the conformational epitope are recognized mainly by the B cells. The linear epitopes are recognized mainly by the T cells but can be recognized by the B cells. Once again, شو الفرق بين اللينير epitope والconformational epitope؟ اللينير epitope بيكون في عندنا الترتيب of the residues في نفس المنطقة، متتابعين على نفس المنطقة. لما نحكي عن conformational epitope زي ما انتم شايفين due to the folding of the protein، البروتين لما يصير له folding بيعمل لنا هذا التجويف. بس هذا التجويف هو لا يشكل سيكونس متتابع إنما متفرق، وبالتالي once بصير في denaturation لهالبروتين هذا راح تنفصل هذه الأجزاء عن بعضها البعض، وبالتالي it is no longer antigenic لأنه هو الانتيجينيسيتي جاية من كونهم قريبين من بعض وماخذين شكل معين. إذا بصير denaturation وبتفكك البروتين وبنعمله linearization راح تبعد المناطق عن بعض، وبالتالي ما راح ترجع تشكل منطقة antigenic. We have three types of antigens. We have exogenous antigen from an outer source, we have endogenous antigen from an inner source, and we have autoantigen from self. So the exogenous antigen are antigens that have entered the body from the outside, for example, by inhalation, ingestion, or injection. So that they are from an outer source. We have bacterial antigens, we have viral antigens. An example of bacterial antigens, we have antigens that are related to bacterial cells, such as the O antigen, the somatic antigen in bacteria, the capsular antigen, the flagellar antigen, etc. Also, we have antigens which are secreted by the bacteria itself. So they are not related to the bacterial structure or cell, and they are produced or secreted by the bacteria, such as the exotoxins. Viral antigens, we have the structural viral proteins, which are the capsid protein and the glycoproteins on the surface of the enveloped viruses. Also, we have soluble antigens, such as soluble nucleoproteins, which are an example of viral exogenous antigens as well. Endogenous antigens are antigens that have been generated within cells as a result of normal cell metabolism or because of viral or intracellular bacterial infection. So they are formed as a result of normal cell metabolism or as a result of intracellular infection caused by viruses or bacteria. Examples of the endogenous antigens, we have the blood group antigens. The A, B, and Rh antigens are examples of endogenous antigens. We have the histocompatibility antigens. They are glycoprotein molecules present on all nucleated cells. We have the major histocompatibility complex antigen, and we have the human leukocyte antigen, and those are an example of an endogenous antigens. And once again, any intracellular infection, whether with viruses or bacteria, represents a source of an endogenous antigen. Autoantigens are usually a normal protein or complex of proteins, sometimes it might be DNA or RNA, that's recognized by the immune system of a patient suffering from a specific autoimmune disease. So usually the immune system does not recognize the self-antigen as foreign, and it does not target them, and it does not form antibodies or target them with the T cells. But in certain instances, autoimmune disease occurs, so the immune system might target the self proteins. These antigens, under normal conditions, not to be targeted for the immune system, but due to mainly genetic and environmental factors, the normal immunologic tolerance for such an antigen has been lost in these patients. زي ما حكينا المرضى اللي بصير عندهم autoimmune disease بصير جهاز المناعة يهاجم الجسم نفسه، خلايا الجسم نفسه، وهي بتحصل due to genetic or environmental factors. Superantigens. Superantigens, they activate multiple clones of T lymphocytes. They are active at very low concentration, causing release of large amount of cytokines. The massive T cell activation and release of large amount of cytokines cause systemic toxicity. It does not lead to acquired immunity. There is no memory. Example, في عنا الستاف aureus toxic shock syndrome toxin and enterotoxin، وعنا example اللي هو الstrep pyrogenic toxin A. Those are two examples of superantigens مروا معنا وحكينا عنهم قبل هيك. شو مواصفات السوبر انتيجين؟ مواصفاته it activates multiple clones of T lymphocyte، وبالتالي احنا حكينا اذا في عندنا انتيجين بالعاده بعطينا ايش؟ It's going to be recognized by T cell B cell اللي هي equivalent to that antigen، يعني بتكون specific. يعني اذا الانتيجين X، البي سيل او التي سيل اللي عليها الكود تبع انتيجين X is going to be activated. بس لما نحكي عن السوبر انتيجين ما بنحكي عن نوع واحد من البي او التي سيل، بنحكي عن التي سيل سوري، بنحكي عن multiple clones، مجموعه كبيره مختلفه الانواع من التي lymphocytes are activated. والاكتيفيشن بتصير حتى على very low concentration، يعني very low concentration من الانتيجين هذا او هذا is going to elicit T cell activation. Also, it's going to lead to release of large amount of cytokine. These cytokines are going to further activate the immune system، وبالتالي بصير في عنا hyperactivation of the immune system, which might eventually lead to systemic toxicity. لأنه حكينا دائما we need balance in the immune system. Hypo reaction or hyper reaction is going to cause destruction more than benefit to the human body. وبالتالي في حالة السوبر antigen it's going to activate multiple clones of the T lymphocyte at very low concentration. It's going to lead to a release of large amounts of the cytokines, and this is going to lead to systemic toxicity. All this immune system response is not going to lead to formation of memory cells against this toxin. What are the requirements of T cell response? So in order to get a good T cell response, the T cell present mainly in lymph and lymphoid organ. حكينا لما حكينا عن الليمف عن ال organs of the immune system، حكينا إنه T cells موجودة وين؟ zones of the T and the B cells موجودة في الليمف نود، موجودة في السبلين. حكينا إنه الليمف برجع للليمف نود وهناك بصير له scanning. إذا في عندنا foreign antigens is going to be presented to the B and T cells within the lymph node or the spleen. إذا راجع بالليمف بده يكون بالسبلين، إذا راجع مع البلد بده يكون في إذا راجع بالليمف بده يكون بالليمف نود سوري، وإذا راجع مع البلد بده يكون في السبلين. However, microbes usually enter through epithelial cells like skin, respiratory system, alimentary tract, where T cell number is very low. Accordingly, microbial antigen need to be transported to lymph node to enhance chances of encounter with T cell. وبالتالي microbial antigen need to be transported to the lymph node to enhance chances of encounter with the T cell. لأنه في معظم الانتيجنز بيدخلوا من الابيثيليال cells، وفي منطقة الابيثيليال cells the number of T cells are very low. So T cells cannot interact with complex microbial antigens. Antigens need to be captured, processed, and then presented for cells in a specific way in order to allow interaction. هلا let's assume إنه في عنا antigen. الانتيجين كان بالتيشوز، راح مع الليمف، دخل على الليمف نود. الانتيجين complex antigens، الانتيجنز الكبيرة ما راح ينعمل لها إيش؟ presentation until it is processed. لازم إيش يصير؟ لازم يصير لها تكسير لتتكسر إلى أجزاء أو قطع صغيرة so that they can be presented on the major histocompatibility complex to be recognized by the T cell. T cell responds only to protein antigens and do not respond to other types of chemical antigens. يعني T cell respond to protein antigens. إذا في عندنا polysaccharides, lipoproteins, it does not respond to these antigens. طب في الحالة هاي who's going to respond؟ البي سيل. شو الفرق اللي بيعمل لنا اياه اذا الانتيجين كان بروتين ولا مش بروتين؟ متذكرين لما حكينا عن الفاكسينز خاصة اللي بينعطوا للأطفال أقل من سنتين و they found they were not effective، وجدوا إنه الحل إنهم يعملوا conjugation حتى يقدروا يعطوهم للأطفال سنتين. الجواب في هذه السلايد. حاولوا تتذكروا شو هم الفاكسينز that were not effective if given to children below 2 years unless conjugated. Antigen presenting cells, a group of immune cells whose role is to take up, process and present antigenic peptides to a cell. Antigen presenting cells من اسمها هي خلايا تقوم بعرض الفورين antigens على جهاز المناعة in order to be recognized as foreign and to elicit immune response to eradicate and eliminate the invading pathogen. We have professional antigen presenting cells and we have non-professional antigen presenting cells. Professional antigen presenting cells are the macrophages, dendritic cells, and the B cells, which can express MHC class II molecules. Non-professional antigen presenting cells, other cell types capable of expressing MHC class I molecule. A class II molecule. And the endothelial cells, the fibroblast, will activated T cells. So the antigen presenting cells, we have professional, non-professional. Professional, macrophage, dendritic cell, and B cells. And they express MHC class II molecule. Non-professional, we have the endothelial cells, the fibroblast, and the activated T cell, which also express MHC class II molecule. When we talk about antigen presentation, we need to talk also about the major histocompatibility complex, because both of antigen presentation and major histocompatibility complex work together, because antigen presentation in this professional antigen presenting cell is going to be displayed on the surface of the antigen presenting cell, but it's going to be sitting on the major histocompatibility complex. Major histocompatibility molecules are membrane proteins on antigen presenting cell that display peptide antigens for recognition by T cells. So they are membrane proteins display the peptide for recognition by. لما الفورين antigen يدخل على الخلية حكينا لازم يتقطع إلى أجزاء صغيرة بعدين بصير له ايش؟ بيركب اذا بدنا نسميه كرسي اللي هو الميجر histocompatibility complex، و it's going to be displayed on the surface of the antigen presenting cell. الانتيجين بيكون قاعد على الكرسي اللي هو الميجر histocompatibility complex. Major histocompatibility complex molecules are the principal determinants of acceptance or rejection of tissue graft. It plays an important role in the acceptance or rejection of the tissue graft. We have two types of MHC class, two types of MHC classes. We have MHC class 1 and we have MHC class 2. MHC class 1 plays a role in regulation of immune response to intracellular parasites such as the viruses. All cells can be infected by virus, therefore cells express MHC class. All cells express MHC class 1. The MHC class 1 is expressed in all nucleated cells. The MHC class 2 is expressed on antigen presenting cells. بالتالي كل الخلايا فيهم ايش؟ MHC class 1. بينما ال MHC class 2 هو موجود على خلايا متخصصة اللي هي الانتيجين presenting cells. So this is the MHC class 1 and we can see it is composed of 1 alpha, 2 alpha and 3 alpha chains and 1 beta chain. So the protein structure is composed of 3 alpha, 1 beta, okay? And it's embedded within the transmembrane region of the cell. It contains a peptide binding cleft that accommodates 8 to 11 amino acids. So here we have this peptide binding cleft، المنطقة اللي بيرتبط فيها مع الانتيجين اللي موجود بالعادة حكينا وين؟ داخل الخلية. داخل الخلية. وبالتالي viral infected cell، الفيروس اللي داخل الخلية راح يتكسر إلى أجزاء صغيرة بحدود 8 إلى 11 amino acid which is the most antigenic part of the virus. بيجي هون let's say a glycoprotein 8 إلى 11 amino acid. It's going to be displayed on HC class 1 اللي هي وين؟ on the surface of the cell. And it is going to be recognized by CD8 T cell and activated. As a result, this virally infected cell is going to be targeted by the cytotoxic T cell. Perforins and granzymes are going to be produced, and this is going to lead eventually to killing of this infected cell. Next, we're going to talk about the MHC class 2. We have the MHC class 2 is present only to is present and is restricted to antigen presenting cells. It regulates the immune response to exogenous antigens. إذن في MHC class 1 عم نحكي عن endogenous antigens, endogenous antigens which are present within the cell. لما نحكي عن MHC class 2 نحكي عن exogenous antigens مصدرها خارجي. طيب كيف وصلت لداخل الخلية؟ وكيف صار لها presentation على antigen presenting cell؟ حكينا الانتيجين presenting cells اللي هم زي ايش؟ زي macrophages, the dendritic cells, and they are capable of phagocytosis. وبالتالي exogenous antigen is phagocytosed, internalized into the antigen presenting cell, and then it is lysed in order to be displayed on the major histocompatibility 2. The binding cleft of the major histocompatibility 2 is larger than the binding cleft in major histocompatibility 1. هناك كان في عندنا 8 إلى 11 amino acid. Here we're talking about 10 to 30 amino acids. So the binding cleft in MHC class 2 is larger, and the structure is different. Here we have 2 alpha and 2 beta, 2 alpha helices and 2 beta chains, which make up the structure of the major histocompatibility 2, and they are also embedded within the transmembrane region. So once the antigen is the exogenous antigen is displayed here is going to be recognized by the CD4. So here we need to remember the rule of 8. MHC class 2 is going to interact with CD4, 4 by 2. In the major histocompatibility complex 1, the antigen displayed on MHC class 1 is going to be recognized by the CD8, 8 by 1 or 1 by 8. وبالتالي it sticks to the rule of 8 that we talked about previously. Features of peptides binding to MHC molecule. So each MHC molecule displays one peptide at a time. Each MHC molecule displays one peptide at a time. Peptides are acquired during intracellular assembly. Low affinity and broad specificity binding, so many different peptides can bind to the same MHC molecule and even can bind self-peptides. Low affinity and broad specificity، وبالتالي جاذبية الأنتيجين لل MHC is low، وبالتالي هذا يعني سهولة ارتباطه وانفكاكه عن ال MHC. and broad specificity معناته multiple antigens can be presented on the MHC molecule. Very low off rate. MHC display bound peptides long enough to be located by the T cell. Very low off rate. معناته إنه لما يرتبط once إنه ارتبط the antigen is going to stay for a long period of time, enough for the T cell to see it, recognize it, and start to elicit an immune response against. وبالتالي once ارتبط مش سهل إنه ينفك، بظل لفترة كويسة حتى in order to be recognized by the T cells. Stable expression of MHC molecules require peptides displaying. Stable expression of MHC molecule requires peptide displaying. The MHC molecule bind only to peptides, protein antigens, so T cells can only respond to protein antigen. نفس النقطة اللي حكيناها سابقا. The T cell response is targets only protein antigens. Steps in antigen preparation for T cells. Antigens must be prepared in order to be recognized by the T cells. So this process has 5 steps. The first step is transport. Antigen must be transported to the lymph node for proper interaction with the T cell. So if the antigen starts in the tissue, it's going to go back with the lymph into the lymph node until it reaches the B and the T cell region. And then once the foreign antigen is encountered, is going to interact with the T cells. Second, uptake, access of native antigen and pathogens to intracellular pathways of degradation. واللي هي زي ما حكينا إذا عندنا exogenous antigen كيف بده يوصل داخل الخلية ويصير display على major histocompatibility complex? Phagocytosis. It's going to be uptaken by phagocytosis into an endocytic vesicle. Then in the endocytic vesicle, it's going to be lysed into smaller antigens and degraded. Third, degradation, limited proteolysis of antigens to peptides in order to be of a size that can be displayed on the major histocompatibility complex. Then antigen major histocompatibility complex formation. The fourth step is loading of the peptides into the MHC molecule. And the fifth step is the antigen presentation, transport, and expression of the peptide MHC complex on the surface of cell for recognition by T cell. اللي هي حكيناها وراح نشوف بالسلايد اللي بعديها اللي هو display of antigen, whether it is endogenous or exogenous. So here we have an exogenous antigen. Exogenous antigen is going to be presented on the MHC class 2. Endogenous antigen is going to be displayed on MHC class 1. Exogenous antigen هاي الخلية شو بدها تكون؟ بدها تكون antigen presenting cell. الانتيجين presenting cell ايش عملت؟ عملت phagocytosis. اخذت الانتيجين هذا into phagocytic vesicle، بعدين صار في عندنا degradation of the antigen into smaller residues that can be loaded onto the major histocompatibility complex, produced in the AR, and then interaction between the antigen and the major histocompatibility complex occur, and then this is going to be displayed on the surface of the antigen presenting cell. هاي حكينا ايش antigen presenting cell. This is exogenous pathway. The presentation is going to be on class 2 MHC molecule, and it is going to be recognized by the CD4 T cell. When we talk about endogenous antigen, endogenous بمعنى ايش؟ بمعنى خلية infected by a virus. The virus is inside the cell. Then the mode of lysis, degradation or destruction is the proteasome. Proteasome is going to lyse the viral protein into smaller proteins, and then these proteins are going to attach to the MHC class 1 within the endoplasmic reticulum, and then it's going to be contained within this vesicle, and then it's going to be displayed on the surface of the cell. MHC class 1 is present in all nucleated cells. So this is a normal cell infected with a virus. The difference here is the lysis occurs by proteasome. Here the lysis occurs by degrading enzymes. Here the binding of the foreign antigen to the MHC class 1 occurs within the ER, and then it is exocytosed to the surface of the cell on the MHC class 1, and it is going to be recognized by the CD8. Here, as we said, the destruction or degradation occurs via lysosomal enzymes, and the binding of the foreign antigen to the MHC class 2 occurs outside the endoplasmic reticulum, and then it is displayed on the surface of the antigen presenting cell, and it's going to be recognized by the CD4. So first, antigen capture and transport to lymph node. So immature dendritic cells in the epithelium capture microbial antigens and leave the epithelium. In the epithelium, we have the dendritic cells. The dendritic cells migrate to draining lymph node, being attracted by chemokines produced in the node. During their migration, the dendritic cells mature. Once at the lymph nodes, the dendritic cells start the process of presenting the antigen to the T cell. Dendritic cells at their maturation express different membrane proteins. For example, immature dendritic cell express surface receptors essential for microbial binding and capture, while mature dendritic cell express MHC molecules necessary for antigen processing. طيب نبلش من النقطة الأخيرة. هلا dendritic cells اتفقنا انها professional antigen presenting cells. dendritic cells الموجودة في الابيثيليوم واللي حكينا most of the foreign antigens enter through the epithelium, it's going to be ايش؟ engulfed أو taken up by the dendritic cells. هلا dendritic cells الاماتشور dendritic cells بيكون ايش؟ الاكسبريشن أو receptors اللي عليهم هي receptors لها علاقة في binding and capture of foreign antigen, bacteria, virusat or whatever. Once it captures these viruses, foreign viruses or bacteria, it starts its journey with the lymph to the lymph nodes. And this journey is guided by the chemokines which are produced in the nodes, in the lymph nodes. During this journey, the dendritic cells start to maturate. هلا في المرحلة الأولى في عندهم receptors to attract microbial binding, the viruses, bacteria. بعدين بالطريق they start their maturation and they start expressing the MHC molecules, لأنه في المرحلة التالية لما يدخلوا على النودز in order to present the foreign antigen لل T cells في الليمف نود بلزمهم ايش؟ بلزمهم MHC molecule، وبالتالي they start expressing the MHC molecule. They load the foreign antigen onto the MHC molecule and display it on the surface of the cell to be recognized by the CD4. So here we have the antigen capture. Then activation of the dendritic cell. حكينا في المرحلة هاي بيكونوا expressing receptors that has to do with the capture of pathogens. Then it goes migration of the DC. It goes to the lymph node. Maturation during this journey, it mature to and start displaying or expressing the MHC molecules. And then it's going to display the foreign antigen on the MHC class 2. So the mature dendritic cell presenting antigen to the naive T cell, and then successful process of antigen presentation and recognition by the CD4 cell occurs. Uptake of antigen. The process of entering the antigen into the cell, mainly antigen presenting cell. This is important for exogenous antigen processing, while endogenous antigens are already inside the cell. حكينا الexogenous بصير لهم ايش؟ phagocytosis, and they are engulfed by the antigen presenting cells. Uptake by immature dendritic cells. في عنا ثلاث طرق. في عنا pinocytosis, receptor mediated endocytosis, and phagocytosis. Uptake also by macrophages are governed by the same three processes: phagocytosis, pinocytosis, and endocytosis. Pinocytosis is used usually for liquid or very small molecules or granules. Phagocytosis usually is directed towards large molecules or microbes or complex molecules. And we have the receptor mediated endocytosis. And there are different types of receptor mediated endocytosis. We're going to see them in the next slide. So the uptake of exogenous antigen might occur through phagocytosis for large molecules, might occur through pinocytosis for small molecules or liquids, and might occur via receptor-mediated endocytosis. So we have the FC receptor-mediated phagocytosis. So after the foreign antigen, as the one seen here, is recognized by the antibody, the FC portion of the antibody might bind to the FC receptor, and this might lead to uptake of the foreign antigen via FC receptor mediated phagocytosis. We have also the antigen-antibody complex is going to elicit the complement system activation, and as a result, this is another method of uptake of exogenous antigen through the complement receptor mediated phagocytosis. Also, we have the membrane Ig receptor mediated uptake. So we have phagocytosis, pinocytosis, and receptor-mediated endocytosis. The receptor-mediated endocytosis, we have the Fc receptor-mediated endocytosis, the complement receptor-mediated endocytosis, and the membrane Ig receptor-mediated uptake. The uptake mechanism direct antigen into intracellular vesicle for exogenous antigen processing. وبالتالي هاي العملية كلها عم نحكي عن exogenous antigen, which is going to be captured by antigen presenting cell, which is going to be displayed on major histocompatibility complex 2, which is going to be recognized by CD4 cell. Antigen processing must be processed in order to be recognized by T cells. Require two main steps. The antigen processing, degradation of externally or internally derived antigen into short peptide sequence, and the association of the peptide with MHC class. So الانتيجين processing حكينا بتكون من خطوتين. أول شيء تكسير الانتيجين إلى قطع صغيرة. لما حكينا عن MHC class 1 حكينا عن 8 إلى 11، MHC class 2 حكينا عن 10 إلى 30 amino acid. After that, association of the peptide with the major histocompatibility complex molecule. We have two antigen processing pathways. MHC class 1 for endogenous antigens, MHC class 2 for exogenous antigens. Processing machinery in the MHC class 1, the destruction of the antigen occurs via proteasome. In the MHC class 2, it occurs via lysosomal enzymes. Where do we find the MHC class 1? It's found in all nucleated cells. The MHC class 2 is restricted to professional antigen presenting cells. The site of antigen binding in the major histocompatibility complex 1 protein, it occurs in the endoplasmic reticulum. While for the major histocompatibility class 2, it occurs outside the endoplasmic reticulum within the lysosome and the endosome. The MHC utilized here is 1, here it's 2, and in the major histocompatibility complex 1, it's presented to the CD8, the CD8 T cell, and the major histocompatibility complex class 2, it's presented to the CD4 T cell. The fourth step is antigen presentation, the activation of the T cell via T cell receptor, which specifically recognize antigenic peptide in association with either MHC class 1 or 2 molecule on the surface of antigen presenting cell. واللي حكينا بتصير في عندنا ارتباط الانتيجين مع ال MHC class 1. بعدين ال MHC class 1 is going to be displayed on the surface of the either the antigen presenting cell or any nucleated cell which has an endogenous antigen. The antigen recognition. The antigen is going to be recognized by and binding to B cell receptor or T cell receptor. In case of B cell receptor, the B cell receptor, as we can see here in the figure, they are membrane bound immunoglobulin on the B cells. So this is the membrane. They are membrane bound, embedded within the membrane of the B cell. They are composed of light chains, heavy chains, and this structure resembles the structure of the immunoglobulin or antibody. And we have here the intracellular signaling binding site as well embedded within the membrane. Immunogenic protein antigen. So the protein antigen attaches to this region, and this region is the variable region, and this region is the constant region. The B cell receptors can also be secreted in plasma as antibodies. The T cell receptor is composed of alpha and beta chains, composed of an alpha chain and a beta chain, which are attached or embedded within the T cell. And there is a groove which binds small peptide presented on MHC on surface of antigen presenting cells. So this is the groove which allows for the binding of the antigen. So an alpha chain, beta chain embedded within the T cell and has a groove that allows for the binding of the antigen, which is displayed on the major histocompatibility complex. And this region is considered the variable region, which binds the antigen, and this region is considered the constant domain or non-variable domain of the T cell receptor. So that's all for our third lecture. Thank you for listening, and please do not hesitate to contact me regarding any questions for this lecture. Thank you so much.